Okay, so welcome to, I think this is the last lecture, right, so semester, so congratulations for making it this far. Today I'm going to be talking to you about what I hope will be quite a fun lecture in terms of content, probably something a bit different to what you might have come across before. And it's probably going to tie together a lot of the things that we've been teaching in terms of neuroscience from PHAR3310 and PHAR3320, which is photopharmacology. So as the name suggests, it's the use of light in pharmacology to control drugs. So I'll start off with, why do we actually need photopharmacology? Why is just taking pharmacology and the medicines that we have available, why is that kind of limited? So as you probably know, like through your studies of pharmacology, selectivity is probably one of the biggest issues facing pharmacology. Because when we give drugs or compounds, we need those drugs to be specific, right? We want them to target only the kind of tissue or receptor that we want them to target. And ideally, then whatever compounds we give won't have any effect on any systems outside or say drug targets outside of what we're actually trying to target. But realistically, when we give compounds and drugs, selectivity is never really achieved because we have kind of like off-targeting. So when we give drugs, for example, if we want to target, say, calcium channels in the brain, right? You take a calcium channel inhibitor, that will go into your system and it will inhibit calcium channels anywhere in the body. So we affect multiple targets, we affect receptors that we want, but they're also in organs outside of what we're trying to target. And poor selectivity then leads to a whole range of side effects. So, when we talk about things like anxiolytics and antipsychotics, some of those things, as Jamie probably talked to you about, have very wide side effects. So, for something like anxiolytics, we can get very bad, like gastrointestinal side effects because the receptors we're trying to target are also expressed in the gut, right? So, selectivity is one of the main and off-target issues, and one of the main things that limit our use of pharmacology. And so ideally, one strategy to kind of control selectivity would be if we had a way of actually controlling where does the drug activate, and ideally also where it goes. And photopharmacology can address really where the drug is activated. So we can do this because we can use photopharmacology to actually control the spatial and temporal resolution of drugs. And so by that, I mean by spatial resolution, we can control in space, in X, Y, Z, where the drug is being active. And temporal resolution, we can control how long this drug will be active. So normally, for example, if you take a drug, you obviously need to absorb the drug has its effect, it needs to be metabolized before that effect is over. Photopharmacology allows us to kind of skip that and just go, okay, we want this drug or compound to be on and off, and only when the light is on and off. And I'll explain to you in more detail how that actually happens. So the principle of photopharmacology is really the culmination of pharmacology, chemistry, and physics all coming together. And basically, it's the use of light to control bioactive molecules. And we can use light to kind of change the structure of these compounds when we put the light on them. And light is really great because we can obviously turn light on and off, so we can make these things like control in time. The compounds we're turning on and off, depending on how we use them, can be both irreversible or reversible, so we have very tight control on that. And we can use light to either activate or inactivate drugs and molecules, right? So we have this really wide level of control, provided we can get light in there. So when we use photopharmacology, we need to use light. And why do we want to use light as opposed to, say, sound, right? And I do want to say that sound is actually what do we call, acoustic pharmacology, I guess, is like the use of sound control pharmacology. That's also a really rising field. But light is really great because there are a lot of very light-sensitive molecules out there in nature that we can manipulate. And we can also make these kind of compounds in the lab, compounds that respond to light. Light is so great because the physics of light and the properties of light have been well understood for a very long time, right? So we really know the mechanism of how these things work. We can control light, with great temporal and spatial resolution, so we can use lasers if we want high energy, want really high focus. We can use something just like LEDs. Also has great control of time, but LEDs, provide light over large spaces. So, we have the physics worked out, and we have the equipment to do it. So it's a really great tool. And to give an example of things that are light sensitive, there are a lot of animals like jellyfish that make light sensitive proteins. And we can do, and I'm not sure if Jamie's talked about it, but we can do things where we can take light-sensitive proteins from different species and put them into tissues and control their activity. And I'm going to talk about how we use light to control compounds, and then how we can use that in the nervous system. So there are many different modalities of photopharmacology, and I'm going to go over kind of broadly the main ones, which you need to understand broadly what they are, and then I'm going to focus on one specifically that you really need to understand the mechanism on, and I'll talk about those in great detail. So we can use light to control receptors, ion channels, GPCRs, transporters, etc. And this control could be reversible or irreversible. So to start off with, I'll tell you about how we can use it to make an irreversible control. So the first modality is what we call irreversible inactivation. So here, imagine that we have, for example, a receptor, this thing that kind of looks like a kidney bean, I guess. And we have a ligand or some kind of agonist shown here in the red star. Okay? And so this arrow here is showing you the direction of the reaction. So this, because it's only going one direction, tells you this is only a unidirectional change. In physics and chemistry, HV denotes light. So whenever you see HV on the arrow, this is showing you that light is being applied to get this kind of direction. So for example, let's say we have a receptor, it's got its ligand or agonist in the system. It would normally bind to the receptor to have its effect, right? Now, let's say you wanted to turn that effect off or stop that effect from happening to begin with. You can apply light to the system, and this photosensitive or light-sensitive agonist or ligand then changes its structure. So it goes from, say, being a star to a pentagon. So now it can no longer bind to its target, and we no longer have its effect, right? So in irreversible inactivation, light is used to alter the structure of the compound so it no longer binds to its target. So that's the first modality. The second modality is something called cleaving or uncaging, or photocleaving, photo-uncaging. And this is where we use light to kind of break off something so that it can bind to whatever it wants to do. And this might sound a little bit strange. This is the mechanism I'll talk about in a lot more detail. But to summarize, again, we have some kind of receptor or some kind of target. We have its, say, a ligand or agonist that would normally bind to it, this star. But you see that it's bound to something, this circle here. It's being protected. It's being caged by something. We then apply light. This light breaks the bond between the caged substance and the thing protecting it to release the thing we want to bind to its target, and we can have an effect. Right? So in photocleaving and photo-uncaging, the use of light is used to cleave or uncage a molecule of interest from another, and this is used to release a compound so then it can bind to its target. And like I said, I'll go over this in greater detail on how we can use it in the nervous system. So that's irreversible. Now we'll talk about some of the reversible modalities. So in reversible activation or inactivation, this is where the reaction can go two ways, right? And so in this, again, we have our receptor or our target, could be a transporter, whatever. Okay, so we have, say, a compound, a molecule, and depending on the light that we apply, we can change its structure. Okay, so this is what we call photochromic ligands, right? So this is just a fancy term for saying a photosensitive ligand. And they're freely diffusible. So if we put them into system, they're flying around the extracellular space. They could be flying around the inside of a cell, whatever. Okay, so in the first part, we could use one wavelength of light, so a specific wavelength of light. That converts the structure to, say, a star. Okay, so now it's in the right shape. It can now bind to its receptor and have its effect. Okay, so you've turned it on, you've activated it. Now, for example, you want to inactivate it. You want to control when you want to stop that effect from happening. You then apply a different wavelength of light. This then causes the structure of this ligand to revert back to, say, this pentagon, and it can now no longer bind, and you no longer get its effect. Right? So this is a great example of how we can use photopharmacology to kind of turn things on and off. The key thing to remember here is that not just any light will do this, right? So only a light of a specific wavelength will cause the reaction this way, and only a specific wavelength of light will do it this way. And it's important that these things are controlled by different wavelengths of light, because if you have them overlap, the reaction will go, both ways. So you need very tight control on two different wavelengths. So this other one is called tethering. And so tethering is when a ligand is actually tethered to its receptor, as shown in this diagram here. And what you do is, if you apply a light, you can actually change the binding efficacy for that ligand, which is tethered, to bind to its receptor or Okay. So when we have, say, light on in this case, we convert the structure such that it can now increase its binding efficacy to bind to its receptor, and we can have its effect, right? So it's very simple to remember in terms of like it's a bidirectional. We can turn it on and off. But the thing with tethering is, like, think of it as simply, it's like the ligand is literally tethered to the receptor, and the light is basically changing the binding efficacy of whether it will bind to the receptor or not. There are two types of tethering. This one is kind of really boring, so I'm not going to go into great details. But basically, depending on how long you have the tether, will change it. But you don't really need to know that. You just need to know the basics of this system here and understand how it works. And so lastly, we have something called cross-linking. So again, we might have, say, a receptor, or it might be, say, a good example would be like a transporter or ion channel. Okay, so let's say we have an ion channel shown here, and we have this kind of light-sensitive switch shown here, okay, which is connected to the ion channel. Think of it as, say, for example, being bound to like both sides of a pore. Now, if you put light on it, you can cause a conformational change in the structure of it, and then if that changes the structure, that'll physically like pull or open the thing it's attached to, right? So, for example, let's say we have, say, an ion channel here. The tether is, say, keeping the ion channel open. If we apply a light, that causes this structure to change to, say, bring it closer together, so that, for example, that might close the channel, and we have an effect, right? And the light can be used to turn that on and off, right? So cross-linking, think of it as a physical structure linking like part of the structure together, and depending on the light, we can say open or close something. Okay. Is that all kind of making sense? Yeah. So again, as long as you understand the different modalities and basically how they work, that's what you need to know. This slide is basically, I'm trying to show you that we have lots of different kind of like photosensitive compounds that we can use to control different aspects of the nervous system. So for example, we have types of photo switches for ion channels. You'll notice with all of these things, they have really terrible names, so don't remember any of the names. You don't need to know what names bind to what. I'm just trying to show you here that we have some of these substances, and we are using some of these substances to control things like AMPA receptors for ion channels. We can target, like, for example, GAT1, which is a transporter for GABA. We have, hopefully remember, EAAT, which is the transporter for glutamate onto glutamatergic cells. But we also have photo switches for GPCRs. For example, when we talked about the CB1 receptor the other day, we can control some of these receptors. But again, don't worry about the names and what they do. It's just to give you an example. So the main one that you really need to understand in like a bit more greater detail is photo-uncaging, because we're going to talk about how we can use that in the nervous system. So in photo-uncaging, we have these cage compounds, okay? And cage compounds are compounds that are kind of like encapsulated or caged by light-sensitive protective groups, okay? So here we have our cage compound, and this is our protective molecule. And in the protecting group, shown here, right, it's actually got this photo-removable chromophore. And so for those of you who may not know what chromophores are, I've just put the definition here, okay? And these chromophores are bonded to the molecule of interest, so for example, the cage compound, okay? So what happens in photo-uncaging is that we apply light, okay? That light has to be a very specific wavelength. That light is absorbed by the chromophore, and when that happens, the two structures separate, okay? Photolysis, as shown here. This uncaged molecule can then bind to whatever it needs to do, right? This thing kind of looks like an olive, but it's not, so it's actually a receptor. So for example, we have a cage molecule, light comes in, the chromophore absorbs that light, breaking the bond between the protective group and the cage group. They separate, go their own two ways, and now the caged group can bind to whatever its target is. Okay? And in neuroscience, we will often use this to remove or uncage neurotransmitters. And I'm going to show you what I think are some really cool examples of that. So in principle, very simple, okay? But again, it's very important that you understand how photo-uncaging works. This is just another diagram to illustrate that and kind of like reinforce that learning. So here we have the caging group or the protective group, and this is our biomolecule, our like cage compound of interest. We can cage it, so this is caging the molecule. In its cage, when it's bound to its protective group, it cannot bind to its receptor. All right? So we apply some light. The chromophore absorbs the light, breaking the bond between these two substances. Okay? So then this goes away. This molecule can now bind to its receptor. When we use it in neuroscience, like I said, we often use it to uncage neurotransmitters, and it's probably the most common application of photopharmacology in neuroscience. And we can make cage compounds so that they bind to specific receptors. So we have cage compounds that, for example, bind to NMDA receptors or just AMPA receptors. And these diagrams are really cool because basically these diagrams are showing you what the cage group are and what the protective group are. So here in blue, this is the cage group that we're trying to break off. This is the thing that's actually going to bind to our receptor. And you can see that it's bound to its protective group here, shown in white. Okay? So when we have that light come in to break them off, it literally breaks them at these bonds here, right? And that's how they separate. All right, cool. So I want to show you a really neat example of how this was done, all the way back in the 90s. Some of you may or may not have been born at that stage. But this is just an example. Hopefully you've seen this diagram before. Has Jamie shown you a cartoon of these neurons before? Yes? Okay. So here we have a neuron. You see these little like spiny kind of like mushroom-looking things coming off the cells. These are actually what we call dendritic spines. These are actually the postsynaptic structure, right? So we would normally have a axonal terminal coming to connect to each one of these. Each one of these represents a neuronal connection. And so the postsynapse will have things like glutamate receptors. And so back in the 90s, it was shown that, if we have these cage compounds, this is actually a real-life example. This is a neuron in actually a mouse brain slice. So this neuron is living. It's being perfused with oxygen and all the salt solutions it needs. And what they're going to do is they're going to put the cage compound all around it. So it's just flying around, it's floating around the neuron. And what they do is they apply a laser at very specific parts of the neuron to target individual synapses. And what they do is they're recording the electrical activity through this pipette, through this electrode, into the cell body, shown here. And so what they've done quite nicely is they've shown us, okay, if we shine light, a laser, on individual synapses shown in the numbers here, what is the glutamate response, right? Because when we shine the laser here, only glutamate is uncaged at that point, at that synapse. And what you can see is that if you start uncaging glutamate at different synapses, you'll see that the response is actually quite different. Right? And this tells us then that synapses, they don't all have the same density or type of glutamate receptors, because if they did, then the response shown here in white would be exactly the same. And so using this technique, we can start to study, what parts of neurons have what type of receptors, what is their density, how fast are the kinetics of these receptors and channels. So this is a really elegant demonstration of how it was done back in the 90s. This is an example of more recent of what we can do today. So this paper is from 2011, but we still commonly do this today. So we can do photo-uncaging in animals which are awake, or organisms that are awake and living. So we can study how living neurons respond to photo-uncaging. So here we have a diagram of a mouse. The mouse has had part of its skull actually replaced with a glass cover slip. And what you can do then is you can put your same electrode into a living neuron, so we can record its activity. We can give that animal a photo-uncaging compound, or we could just put it directly onto the brain, doesn't matter. And then what we can do is we can use light and shine it onto different aspects of the neuron, and record what the glutamate response is to different parts of neurons. So here's the glass window. Here's the neurons being recorded. It's being filled with a fluorescent dye, which is why it appears white. Here we have the branch of the dendrite. These little knobbly things are our postsynaptic our spines. They're the postsynaptic structures. So like that single dot here, that's a single synapse. And so what they're doing is, again, with very fine lasers, is shining the light onto individual synapses and recording what is the glutamate response at these individual synapses. And what you can see here again is that the response to glutamate after it's being uncaged is not the same across a neuron. Even neurons which are almost next to each other, they show completely different responses. Okay, so this neuron, this synapse here at 3 basically shows a very minimal response, whereas the synapse at 1 shows a very large response to glutamate. So another example of how we can use uncaging to tell us, what are the densities of these receptors at synapses? Are they the same kind of receptors at different synapses? This, I think, is a really interesting experiment. And here we have, again, the branch of a dendrite. This is the cartoon. This is real life. This is the postsynaptic structure. So this is dendritic spine. You'll see here, there's no dendritic spine here, right? So there's no synapse here. But what this experiment did was, okay, if we uncage glutamate on a part of a neuron that doesn't have a synapse, what actually happens to it? And what was quite amazing is that if you uncage glutamate on, say, a glutamatergic neuron, so these neurons do actually express glutamate receptors, a synapse actually starts to grow on these neurons. And so here's another example. Here's a dendritic branch. There's no synapse here. If they uncage glutamate here, a synapse appears on it, right? Now, this is really cool because it tells us, again, like what parts of neurons have these kind of receptors, what the properties of single synapses. But we can also then start to use photopharmacology to artificially induce things like synaptic plasticity, which is really difficult for me to, I guess, express how wild that is. If you think that, I'm not sure if Jamie talked about it, or maybe I talked about it in semester 1, but like synapses, we think, encode things like learning and memory, right? So we know from neuroscience that when you learn, right now, hopefully you're learning some of this information, the neurons in your brain are changing in that the number of synapses are changing in your brain. The structures are either coming or going. We know that we need to form and remove synapses to form memory. If we can artificially do this, we can artificially, hopefully, control things like learning and memory, right? Now, we're probably way off for exploiting that in a therapeutic context, right? For things like, for example, if we want to improve learning and memory for memory disorders. But this provides proof of principle that we can use photopharmacology to manipulate things like learning and memory. This is an example of how we can actually use 2 types of photo uncaging. So here we have a substance that uncages glutamate, and it's only activated with green light. This is actually a neuron that projects up here. This is the cell body. This is the dendrite up here. And we have another uncaged compound, which is only in GABA, but it's only released with red light. So glutamate, as you hopefully know, when you apply glutamate, it binds to its receptors. It causes depolarization, and you get an action potential. Okay, so if we put an uncaged glutamate compound that reacts to green light, we shine the green light, glutamate binds, we hopefully get an action potential. Cool. Now, if we also put in the compound for GABA that only responds to red light, so GABA release should inhibit neurons. We should get hyperpolarization. You'll see that if we put the red light on, the neuron starts to hyperpolarize a bit. The neuron has become inhibited. So even if you try to induce an action potential with the glutamate uncaging, like it induced the AP before, we don't get it anymore, right? So this is how we can use different types of uncaging in a system. We can do that provided that the things that are caged are activated by different wavelengths of light. Because if they both were activated by green light, we would have them both activated at the same time. But the fact that they're only activated by different types of wavelengths, different green or red, we have control on what's being activated, where it's being activated, and how long it's being activated, right? So that goes back to what I was saying before about having tight control on both the spatial and temporal resolution of these things. Okay, so another example is that we could use photocaging to control the release of drugs, okay? So this is a good example of morphine. So morphine, hopefully, I'm not sure if Jamie's talked about it, morphine is good for treating things like pain, okay? So morphine is usually administered orally or by a pump, okay? But again, it suffers from that selectivity issue. It has to be absorbed before it takes an effect. It then has to be metabolized. What could happen if we had control over it with photopharmacology? So here is a compound that is actually caging morphine, okay? So when this is the protective group bound to morphine. When we shine light, we break them off. And so this is an electrical recording from a cell. And you can see that when it's in the dark, so no light is on, the response from the cell is basically nothing, right? If we now have our caged morphine in the system and we apply purple light, in this case, to uncage it, you can see now that it's reacting, right? There's a change in the cell. The morphine is actually able to bind to its receptor. If you just compare, for example, when you give straight morphine, which we know should have an effect, you can see that the photo-uncaged effect is actually very similar to the effect of morphine by itself. It's maybe not as big, but it's pretty close, right? Difference being here is that we have very tight control on how long we want to apply it for. So hopefully, photopharmacology might give us a way of improving selectivity and spatial and temporal control for morphine in the future. This is an example of how we can use photo-uncaging to treat things like cancer. So the details here aren't super important. I'll just walk you through it. But basically, this is our control group in this top row here. This is the dark group, so this is not going to get any photo-uncaging. This is the light group, so this is going to get photo-uncaging. So this is the photo-uncaging drug at different concentrations. And what you can see is, hopefully slightly, is that if you uncage this cytotoxic drug, which is meant to kill the cancer cells, there are less cells, cancer cells, in the group that's received the photo-uncaged cytotoxic group compared to the group that doesn't get light. Okay? That's probably more obvious in this example here, which the drug has been given for longer. The light's been given for longer. This group has the photo-uncaged group but no light, so that the drug is not being uncaged. This group has the light, so it's being uncaged, and you can see that there are less cells compared to control group, right? So photo-uncaging to control drugs is not just something in neuroscience. Hopefully it's starting to expand into other systems. This is just another example, very visually easy to understand. This is cancer being grown in a dish. This is control. You can see that the cancer cells aggregated is quite large. This is the group, same group, but now it's being uncaged with a cytotoxic drug, and the cancer has become smaller. Okay, so potentially maybe we could use photo-uncaging to control drugs that can kill things such as cancer. This is just an example to say I've talked all about controlling neurotransmitter release, but we can also use photo switching to open things like transporters, neurotransmitter transporters. So this is an example of how we could use light. This is not uncaging, this is photo switching, going back to that one of the modalities we talked about before. But we could say have a photosensitive molecule which in this conformation keeps something closed. For example, the GABA transporter closed. We apply a light that changes its structure, it removes it, and now something can go through. Now GABA can go through, right? So as you can see here, this is when it's in dark, in blue, and when the light is on in red, and the GABA reuptake is increased when we have the light on. So just another example, we can also use photo switching to control neurotransmitter reuptake through its transporters. Okay, so to finish off with, like everything, there are advantages and disadvantages to everything. Okay, so one of the great things about photopharmacology is that, like I mentioned, it has great control over the temporal and spatial activity, right? Which we don't have when we give things intravenously or we give it orally. This then allows us to control its off-target side effects, because theoretically we only get this change where we apply a light, and we can control where we give light and how long we give it for. As the technology grows, it's great because we can start to use a combination of drugs and control. So we can turn things on and off, different types of things, like we could say turn GABA on and glutamate off, or we could put one drug in, turn it on, another drug on and off. So we can combine these things as long as they're sensitive to different wavelengths. So that's the advantages. The disadvantages, which is probably something you've already thought about, is like, how do we then get light into the system, right? So, for example, a good example would be Parkinson's disease, right? So currently, Parkinson's disease is treated by deep brain stimulation, where you put an electrode deep into the brain, okay? Now, for example, if you wanted to do the same thing with photopharmacology to release dopamine, you have to get light into the brain, right? And we're not going to shoot a laser into somebody's skull, right, all day, because that light still can't penetrate into the brain. So if we want to deliver that photopharmacology to a human, say for Parkinson's, we would still need to, say, insert something deep into the brain. So the disadvantage is that, you know, to get these things into tissue, you physically have to put them into the tissue if the thing is not, say, on the skin, where light can easily get to, or the eye, right? Photopharmacology probably has a very interesting role in terms of the eye, because light naturally gets in there, and we might be able to do that. One of the other disadvantages is that depending on the substance that you're using, and the intensity of light that you're using and the wavelength of light you're using, you can actually get phototoxicity, right? So some of these wavelengths, if you apply them for too long and too high, they develop toxic metabolites, and that can damage, obviously, the cells, which is counterintuitive to what we want to do, unless, I guess, you want to kill something, like, for example, cancer cells. And as I mentioned before, like, you know, implanting light sources has very difficult technical challenges. So, you know, for example, whenever you insert something into the brain, you usually get some kind of scar formation. So the glial cells usually form a scar around whatever you implant, right? And then if you have this buildup of glial cells or a scar around, say, the thing delivering light, that then prevents the amount of light getting into the brain, right? So just like when we use electrodes for, say, deep brain stimulation, over time the effect will start to decrease, because there will be a scar forming around the implant, which doesn't let the same amount of light to go in, and so we don't get the same effect. I've put at the end here some lecture outcomes, because some people asked me very politely to put them in, so I have put them in. But as I mentioned to the students the other day, generally my lectures are shorter, so like I try to put only the important information you need to understand on them, and hopefully I've made it very clear like what things you need to remember and what things you don't need to remember. So with that, that's it. You guys are free to some extent, and hopefully I'll see some of you at the tutorial next week. But if not, best of luck for the quiz and the exam, and best of luck for whatever you choose to do after this year.